So this is good. So I'm going to put the mic here.
Okay.
Okay.
All right.
Okay, guys, I want to welcome all of you to the Spring 2020 Ocean Science Engineering Seminar Series. Our first speaker today is Jim Morris, and given that Jim is a very close colleague of Joel, I have Joel actually introduce our first speaker. But welcome, and let's get going.
Okay, yeah, thanks for your patience. So we're running a few minutes late. So I'd like to introduce Jim Morris. So Jim Morris is a distinguished research professor from Baruch Institute, which is the marine lab at the University of South Carolina. So he did his graduate work at Yale, and then he postdoced at the Marine Biological Lab at Woods Hole. He's a fellow of the Society for Wetland Scientists and also of the AAAS. Currently, he's a member of the UNESCO Blue Carbon Working Group, so the international body charged with blue carbon. And then also he's on the NOAA National Estuarine Research Reserve Advisory Board. Many of us work in those. And what I always say about Jim is, well, first of all, I've known Jim for a long time and enjoyed working with him. He's an excellent scientist, but also that he's the one that developed the Marsh Equilibrium model, which is used by the U.S. Army Corps of Engineers as well as NOAA to assess the impacts of climate change on marsh ecosystems. And so, Jim, thanks for coming, and I look forward to your presentation.
Okay, thank you. Can you hear me okay? All right. Thank you, Joel, for the invitation, and Manu. I've enjoyed my visit here so far. So I'm going to tell you a story, and the story began in 1984 here. This is North Inlet Estuary in South Carolina, and this is the university's field lab is right here. Actually, it's a great to have a field lab with great analytical chem facilities right there next to the salt marsh. What a deal is that? So I need to do something. I guess I'm going to have to do this, yeah? No, like that. There we go. Okay, so that's where the estuary is located along the Atlantic coast. It's been there for a long time. I also work in Plum Island estuary, and everything I'm doing in South Carolina, I do in Plum Island, which is north of Boston. And this is a harsh environment. It's hard working here, but it's beautiful. But I only have about 15 years of data, so I'm not going to talk about Plum Island except for one little piece of data that I'll show you. And this is kind of where I'm going with this. This is my conceptual model for how a salt marsh ecosystem works. And I'm going to be talking about different pieces of this during the seminar. Now, this is a really complex microbiome. And Joel's group is far more expert in this than I am. But I've got some notions about how this works and how it interacts with the plant community. These represent flows of things. These represent sort of connections. Sea level affects the growth of the plant. The plant, through its root system, produces acetate, which feeds the sulfate-reducing bacteria that do various interesting things. And there's a complex system of negative feedbacks here, which keeps this system going. And so I'm going to start in the late 70s. I mean, everybody knew that. Everybody knew that salt marshes were nitrogen limited. And for my thesis, I was interested in why that was, because there's a lot of nitrogen in salt marsh soils. And so I did a thesis on the uptake kinetics of nitrogen. And I can tell you why, but I'm not going to do that in this seminar. So this is a bird perch. It's a sign like keep out or something like that. But the birds, ospreys and egrets, rest here, and that's how the plants have responded to the birds that are sitting there. They're obviously responding to the nutrients. I mean, we've known this forever. That's not a mystery. Then by about 1980, suddenly the focus shifted to sulfides. Everybody was thinking that sulfides were toxic to plants, and that was a problem in salt marshes. And so understanding sulfate reduction and sulfides and how it affected the plants, that became kind of important. So I started an experiment in North Inlet with Gary King. Some of you know him pretty well. And Gary King was measuring sulfate reduction. I was doing the plants. And we had this experimental setup where we added an iron mineral to the soil, girthite, and we added this to precipitate the sulfides. And we were, the hypothesis was that if we take the sulfides out, we'll increase the productivity of the marsh. Well, that didn't work. It didn't work because the iron also precipitated the phosphorus. And so we created a phosphorus deficiency. Now, because of the nature of these plots where we were working, we couldn't put girthite over a huge landscape. So we were working at a plot scale. So I had to develop a way of monitoring the productivity of these plants non-destructively so that we could monitor the same site over time, the same sites. And so I came up with this idea of staking out permanent plots, little plots. I mean, these are pretty small plots. They're like 10 by 10 centimeters on a side, larger in other areas. And we tag the plants, the stems, with bird bands so we can follow the individuals. We measure their heights, and from their heights we can measure their weights. Okay, now this is a huge amount of work, and this relationship changes seasonally, so we have to account for this seasonal change. So we started doing this. I never intended that I would do this for more than 3 years, but something really interesting happened right off the bat. On the same plots, from year to year, there was huge variability in the productivity of this marsh. And I was surprised by that because I thought marshes were really predictable environments. Pick up any tide chart, for example. Very predictable. And I looked at everything. I looked at rainfall, solar radiation, you name it. And then I was talking to an oceanographer friend of mine, Bjorn Sharp, and he said, well, did you look at sea level? I thought to myself, well, why would I do that? And I didn't realize it at the time, but there's this solar annual cycle in sea levels. The level of the ocean changes by about 30 centimeters from summer to winter due to thermal expansion of the ocean. Now, because that's not astronomically forced, that's a climate-related or weather-related phenomenon, there's a lot of variability from year to year. So these dots are the monthly means, and so you can see the annual cycle, blah, 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 blah. And the growth of the plants turned out to be correlated positively with the summer sea level. High sea level, high productivity. And so I was motivated to keep doing this. And then after a whole bunch of years, I saw this trend, a very significant trend. And these are 2 different sites now, by the way. This was so amazing, I decided to add another site. This trend is significant. And so I reasoned that, well, if high water level is good for the marshes, if the overall productivity is increasing, then these marshes must not be keeping up with sea level. They're falling behind. They're getting wetter. And so I started measuring the elevations of the marsh. Now, this is from Plum Island. These are productivity data from the frozen marsh in Plum Island. It's not frozen in the summertime. It's pretty productive. And this is a single site, and this is productivity from year to year as a function of mean high water level. And so we see the same thing in Plum Island. So this is not an isolated phenomenon. So I started measuring elevations using these devices called sedimentation erosion tables. Do you guys know what these are? Sets? what you do is you drive a pipe into the ground to the point of refusal. Actually, it's to the point of exhaustion. You drive these pipes down, they're stable. And then this set device, there's a collar that's cemented to the top of these pipes with notches in it. And then you can put this set device down, you level it very carefully, and the notches make it so that every time you put the thing down, you can orient it in the exact same spot. And so when you do this repeatedly, and this is now relative to time zero elevation, when you do this repeatedly, you can monitor the change in elevation. Now, when I started this, I added these fertilization plots. So this is a sort of a split plot design. One half was a control, one half was fertilized. I did this thinking that, okay, nitrogen is going to increase the decomposition in these sediments, and that's going to collapse the marsh. That was the hypothesis. That was wrong. What happened was that the fertilized plots, they took off. The elevations just went, through the roof. And that was an epiphany. I got it. I figured out, that was the observation. That's how the marsh tracks sea level. Okay, when sea level rises and productivity increases, that raises the elevation of the marsh. And so there's an equilibrium that's established between the elevation of the marsh and the rate of sea level rise. And I'll show you more of that as we go on. And these are more recent data from the same sites. We're still doing this. We're still also measuring the productivity on these sites. I have a technician that does this. And so these are the controls. These are the these are the end of year elevations. There's actually kind of a cycle a seasonal cycle in the elevations. So the controls are going up according to the model fit. So this is the marsh equilibrium model, version 5, fitted to these data. And it says that this marsh is growing at a little over 1 millimeter a year. So it's not keeping up with sea level. So I was right. The marsh is not keeping up with sea level. The fertilized sites, they were growing at almost 5 millimeters a year. I stopped fertilizing at this point. I lost funding from USGS and I was pissed off. So I stopped the fertilization. And interestingly enough, the elevations then came down a little bit. We've started fertilizing again. We're going to see how high we can get these things to go. At some point, they have to stop. And interestingly, the loss in elevation here is accounted for precisely by the loss in volume of the live below ground biomass. So when we stopped fertilizing, the biomass decreased. How do we know that? Because I have friends who measure. I'll show you in a minute. So the plants, here's how it works. When the plants trap sediment, the more stems there are, and the higher the biomass, the more sediment they trap. Okay, that's one of the mechanisms. The other mechanism is the growth of biovolume. Now these are cores taken from a NP factorial experiment that has been going on out there. I guess when these cores were taken, they've been fertilized probably for 25 years. So here's a CAT scan. So here's the trick. You have to know a doc in a hospital and sneak these nasty old cores in the middle of the night and run them through this $1 million CAT scan machine. And these are the controls. This is the surface of the core. You're looking down core. You're looking at roots and rhizomes. This is the phosphorus treated core. This is cool in three-dimensional. In 3 dimensions, by the way. You can rotate these things and you can measure the volumes, and therefore you can get at the biomass. This is nitrogen and phosphorus, and this is nitrogen alone. Nitrogen is a big deal. I said that these systems are nitrogen limited. They're secondarily phosphorus limited, but they're primarily nitrogen limited. They make a lot of biomass. And so contrary to what some people are saying today, nitrogen does not decrease below ground production, it increases. Now, I'll show you some more data. So those observations led to the development of the first marsh equilibrium model. And this is the one that's most abstract. I think of it like a late Picasso. It's very abstract. You can kind of still recognize what it is. And it describes the important features of what's going on in a very simple way. And that was the idea. Keep it simple, stupid. So some of the important variables here are depth. So I'll be talking a lot about depth. And by depth, I mean the depth of the marsh platform below mean high water. That's what I mean by depth. And so the change in the elevation of the marsh is a function of depth. You know, if depth is 0, then there's no mineral getting to the surface of the marsh. So it's a function of the depth and a settling velocity turn. And it's proportional to the biomass. So the plant's making biomass. Now that's also related to depth. And so D is depth, QD is sedimentation rate, BS is the surface biomass density, Now the biomass, though, is a parabolic function of depth. There's the plants grow within a range. There's a depth that's too great and they drown. And there's a depth that's too shallow and osmotic stress gets them. It's too dry. And there's a sweet spot in the middle. And you can roughly describe this with a parabola. And so if you substitute this, you can then take biomass out of the equation, and then you've got dz dt is strictly as a function of depth. So this boils down to a cubic, which you can solve, and there are 2 real roots to this equation. And they look like this. This is really, I think this is fascinating. So if you calculate well, let's look at this one first. This is the equilibrium depth. Okay, so this is the depth at which dz dt would equal the rate of sea level rise, given the parameters that are in this model. And you get the 2 roots look like this when plotted against depth. And you see there are actually, for any given rate of sea level rise that's within this range, there are 2. There are 2 equilibrium depths. This one, though, is not stable, and I'll show you why that is in a minute. This side is not stable. This side is. And you can also calculate. You can substitute the depth back into that biomass equation. You can calculate what the biomass is at these different rates of sea level rise, and you get a figure that looks like this. This is pretty neat. That's the unstable side. That's the stable side. Now, what this tells you qualitatively, the equilibrium depth depends on the rate of sea level rise. Consequently, the productivity depends on the rate of sea level rise. There's an optimum rate of sea level rise for primary production, and therefore carbon sequestration. That is a really cool idea. So that's what we got out of that really simple model. Now, the model has evolved over time. It's far more, there are a lot more bells and whistles in it. So to measure, so I got interested in, okay, let's try and measure that response of the biomass to elevation. And I came up with this design here. This is my invention. People call it a marsh organ because it sort of looks like a pipe organ. These are just PVC pipes filled with sediment and planted. And then after some time, we harvest the plants and we measure the biomass. And this is the standing biomass from these things as a function of their depth below mean high water. And, you know, that's what we get. So if the marsh is on this side of this curve, a rise in sea level decreases the depth, which increases the biomass, which increases the sedimentation rate. and an equilibrium is reestablished. But if the rate of sea level rise is too fast, the equilibrium passes to this side of the curve. This is the unstable side of the curve. On this side of the curve, as sea level rises, the productivity declines. And then the marsh basically spirals out of control at that point, being unstable. Now, these marsh organs now, you find them all over the world. And some people have tried to improve upon the design. Oh, here's what they look like in the buck naked. This is what a brand new marsh organ looks like. And this is one way to get them out there to where you want them to be. And Karen Sundberg, my technician, she won the first North American marsh organ race in 2006. That's the only race we've ever had. And she was the only contestant, so she won by default. And anyway, that's what they look like after they've been out for a while. They're planted, they're filled with sediment, and then they're just planted with little tiny stems at the first of the year that we cut out of the marsh and planting these things. So they start out pretty small. Oh yeah, as I said, so people are trying to improve upon the design. This is a Dutch marsh organ here out in the Baltic. And they have these baffles built around this thing so to keep the wave action from killing the things. And so you see the different elevations that they have inside this contraption. This is nuts. And here's a design that they're using near Xiamen, China. They've got the bleacher version of marsh organs in China. So there are more than one way to skin a cat, I guess. Now, I just concluded an experiment where we fertilized these things, the first time they've ever been fertilized. And we've been following some of these pipes for 5 years to see how they grow. And we started 3 years ago fertilizing a group of them. And after 3 years, this is what the nitrogen treatment looks like. That is a massive volume of mostly rhizomes. And the rhizomes are the real story here. Let's see, these are rhizomes. These are fertilized plants after 3 years. These are live rhizomes, dead rhizomes, live roots, and dead roots. So the roots are a pretty small part of the total biomass and the total volume. These are massive. Now, okay, so these are the controls, and these are age 5, age 4, and so on. So we harvest successive pots of these things and then quantify how much is there. And this was surprising, but especially surprising was the response of the fertilized plants. I mean, this is massive. Some of you have probably heard that nitrogen decreases below ground production in salt marshes. It is not true, okay? The difference between these things is eye-popping. And if you plot, here's a plot of just the time series of these. So these are the controls. This is total plant biomass. And these are the controls. These are the fertilized plants. This is total below ground biomass, live plus dead. These are the controls. These are the fertilized plants. This is eye-popping. And if you calculate the annual growth rate, percentage growth rate of roots, rhizomes, and the total, you see there's a big difference. The rhizomes are really the star of the show. But the roots grow faster in the fertilized treatments, but the root shoot ratios go down when you fertilize. That's true. But the root growth goes up, but nothing like the rhizomes. The rhizomes are just through the roof. And I should add that this is net of decomposition, because there's been some production over time that has decomposed. It's net of decomposition. It's also net of herbivory. And we had some herbivory, significant herbivory in these fertilized treatments. The grasshoppers, insects are really smart. They will identify protein-rich plants, and they will attack them. And the grasshoppers attacked these fertilized plants voraciously, and they removed a fair amount of biomass, which a colleague in China, I hope, is in the process of quantifying that. We'll see. So, okay. Now, think about this for a second. You've got an annual growth rate of 100% per year. Now, so the question is, what limits that? You know, if there wasn't a limit of that of some kind, we'd be up to our necks in Spartina biomass. I mean, we'd be growing mountains of Spartina. How is that possible? Well, it's possible because it's self-limiting. So check this out. And you might be wondering, what happens to that growth curve when you fertilize? Well, this is what happens. This is total above ground biomass. These are the fertilized treatments. These are the controls. This is rhizome biomass, fertilized controls. So they grow themselves. They have the ability to grow themselves to a point where their production is limited. Now I'll say another thing that's important about rhizomes. These are perennial tissues. They're perennial organs. They're anatomically like branches on a tree. They have meristematic tissue that grows leaves, just like a tree. And what we call stems are really modified leaves. Now the stems have meristematic tissue in them as well, but they originate from the meristems and rhizomes. They are perennial, get it? They don't turn over rapidly like roots probably do. And their biomass can build up pretty substantially. And you can calculate the volume of these things. This is based on a meta-analysis of over 5,000 soil samples from marshes all over North America. And this is from first principles, this mixing model. K1 and K2, these are the self-packing densities of organic matter and mineral matter. And the K's come from a fit to the mixing model, to this data set. And what it tells you is that the packing density is it's like the bulk density for these different particle fractions. And the difference, it tells you that gram for gram, organic matter occupies an order of magnitude more volume than mineral matter. But this is really handy to know, because now, if you know the inputs, if you know the mass inputs of the mineral matter and the organic matter, you can calculate the volume, and then therefore you can calculate the change in elevation of the marsh. So that's how I do this. That's how the model works these days. Now let me take you through an example, and this is sort of hypothetical. The numbers are not realistic. I have the biomass production of refractory organic matter, which happens to be about 10% of the biomass that's produced. The lignin content of Spartina is about 10%. Now that's what's preserved. The labile part that decomposes is irrelevant. Doesn't add any volume. It doesn't add new volume to the sediment because it decomposes, and it doesn't matter if it decomposes in 1 year or 5 years. It goes away. It's the refractory production that adds new volume. So if you have a curve that looks like this, of refractory production. You can divide by the self-packing density. You can calculate what the accretion rate then would look like given this level of refractory production as a function of depth below mean high water. Okay, now here's this is mean high water right here, and this is deep, and this is above mean high water. These plants will go about 30 centimeters or so above mean high water, and they'll go about 10 centimeters below mean sea level. That's kind of their range. Okay, so how does this work? If we take that curve, the accretion curve, it looks like this. Where's the equilibrium when the sea level rises 0.4 millimeters a year? Well, if the equilibrium is where the rate of sea level rise equals the rate of vertical accretion. And so there are two equilibrium points. There's one there, and there's one there. Okay, but one of these is not stable. Yeah, so this is high marsh, this is low marsh. Now, because of an anomaly in sea level or mean high water or something, pushes the depth from this point to this point, what happens? Well, what happens is that now the accretion rate is greater than the rate of sea level rise. So what does that do? That decreases the depth. So this point migrates in that direction, and it ends up there. It's trapped there. So that's a stable equilibrium point. It's also a strange attractor. And if the point is over here, and due to an anomaly, the depth decreases to this point, well, now the rate of accretion is less than the rate of sea level rise. So what happens? That point just migrates back up to there. So that's a stable point. If you place it at 80, it has to move in that direction. So that's not a good place. This is not a good place to be. Also, think of this. That's a tipping point. And as the rate of sea level rise increases, these points converge and they move like that. So if the rate of sea level rise goes up to 0.95, that one goes there, and that one goes there. This point is still unstable, and this one is still stable, but the marsh starts to lose its, I don't know, I think of it as freeboard. You know, it's like freeboard. So there's not a lot of room for movement up here. Now let's add sedimentation, mineral sedimentation. Now, mineral sedimentation is essentially a linear function of depth. The more water you have on the surface, the more sedimentation you have, and the more depth you have, the more sedimentation you have. So you have to add these two pieces together. So now you have a piece over here. This represents the mineral part of this. And the mineral part, of course, goes on down here. And this is just added. It's additive. So now you have two stable equilibrium points. You have alternative stable states. That one is stable, like I showed you. That one is not, but this one is. A point up here will migrate in that direction and be captured there. A point that is here will migrate over here and be captured by that point. And I think that's pretty cool. So you've got a stable point there, and you've got a stable point there. So think I don't know, sandbar, shoal type of environment, which could, under the right circumstances, become salt marsh. Okay, now here I'm going to get it gets more interesting, I think. So we know I've made the case that the marsh is responding to these anomalies in sea level. These anomalies are seasonal or annual. There's also this fascinating cycle called the lunar nodal cycle. The moon has a little tilt to its orbit, and it takes 18.6 years to rotate from one extreme to the other. And during that cycle, the moon and the sun will exert maximum or minimum gravitational pull on the surface of the ocean. That changes the mean high water level. Doesn't change mean sea level, it changes mean high water level. And this is a graph of mean high water, the mean amplitude of the tide from the NOAA Charleston gauge since about 1920. And there it is, there's that 18.6 year cycle. These are monthly means. And if you take if you look at the extremes of the monthly means, I mean, it's extreme. And here, this is just more contemporary data here. I've taken it up to 2015. 2015 was about the peak of this cycle. This is when you have really bad king tides during this part of the cycle. So now the question is, this is a pretty significant change in flooding that occurs. Question, does the marsh respond to this? Well, it should, if the theory is right. And I took our productivity data, and at least the first half of it looks like this. And there's an 18.6 year harmonic fitted through these data. It's statistically significant. Yes, it looks like the marsh responds. Now, there's a lot of variability in here, but there's a lot of annual variability from year to year in sea level and in the marsh response. And it's just remarkable to me that we can pick this out of this data set. And it's pretty cool. Nutrients. What's happening to the nutrients in the soil when all this is going on? So we've been measuring nutrients, a depth profile of pore water nutrients monthly, going back to about 1990 or something like that. And we use these permanent wells that are installed. This is a breather tube, and there's an insert pipe that goes in, and this is a slotted pipe. The insert pipe here has a serum vial filled with distilled water capped with Nitex membrane. It sits inside this pipe opposite these slots. And so we can then very quickly swap out a set of vials and analyze them. And this is what the data look like. All right, the blue dots here. I'm gonna show you phosphorus. Phosphorus is the cleanest example, admittedly, but they're all pretty significant. Anyway, here's the phosphorus data. There's an obvious annual cycle here, right? Anybody can see that. Here's a great annual cycle. And so step one is to fit a harmonic through that, to calculate what that is on the average. So what I'm trying to do is I'm trying to take the noise out of this data set. I'm trying to pull the signal out of the noise. Step one is the annual cycle. There it is. But obviously, you look at this and you say, oh, gosh, there's more than that. It looks like, it's an 18.6 year cycle. And there it is. There's the 18.6 year cycle. And then when you add the two of those together, you get something that looks like that. Now, that's much more satisfying. But there's something else going on. It looks like the amplitudes of these things also are changing on an 18.6 year cycle. And when you incorporate that into the statistical model, you get something that looks like that. Now, I'm really happy with that. And that's what the final model looks like. So that tells me what phosphate is doing over time. Now, a couple of things about this. The peak is always in the middle of the summer when the plants are growing at their maximum rates, which is nice if you're a plant, because that's when you need those nutrients. Now, and that's what, with a little more current data, that's what the phosphorus looks like. I think it's beautiful. Now, if you can now look at how these things co-vary. And if you plot one nutrient against another over time, you get these. This is called a phase plane plot. And this is sulfide versus ammonium concentrations. And so over time, it does this. So it goes up, and then it comes back down again. That happens on an 18.6-year cycle. And if you plot ammonium versus phosphate versus sulfide, you get something that looks like this. And the fourth dimension here is color. And color is the growth rates of the plants. And so hot is they're growing fast, and everything is going up at the same time. And then the plants cool off, and everything comes back down. That is the 18.6-year cycle. And I'm going to animate this for you. Hopefully well, I'll turn it down if it's too loud. There's a little music. So I didn't tell you, you should have brought your dancing shoes. Oh. How many of you recognize this music? Okay, you've got to be a certain age. Now I'm going to rotate it for you. That's what it looks like. Oh, it looked like a basket, didn't it? No, it's not. How disappointing. Oh well. Anyway, that's what that looks like. Now, let me go on to this one. Now, you might be wondering, well, what do the fertilized plots look like? Right? You were just about to ask that question, right? I'll play this one for you. It's seasonal. There's an 18.6-year cycle, but it's chaotic. There's an underlying determinism. The harmonics in here are deterministic, but I challenge anyone to predict this. It's chaotic. So fertilizing these plots resulted in chaos. And I'll rotate this one in a minute. There. It's pretty wild looking. Yeah, okay. And the other interesting thing about the contrast between these two is that the scales are very similar. It's like fertilizing these things did not result in concentrations off the charts. Everything is pretty much got a similar scale. Now I want to try and explain what I think is going on here. And I'm going to use this photograph taken through the window of a mesocosm that was at the lab outside. This is after a few months of growth. The plants were put in artificial sediment of kaolin and sand, and so the color looked like this. So this is a rhizome coming through here, and these are roots that are starting to come off. Here's a little root here, and there's some ferric iron deposits on those roots right there. That's pretty cool. So look what you have here. You have, there's a root there with some ferric iron. This black stuff, this is iron monosulfide. So this is highly reduced out here. And there's this little halo of oxidized rhizosphere right around here. So what's happening, I think, is that this has got to be from acetate. You always find a lot of acetate where you have Spartina, and the plants are leaking acetate. And the acetate is a perfect substrate for sulfate-reducing bacteria. So they're taking this acetate and reducing sulfate, producing hydrogen sulfide, which reacts with the iron to produce iron monosulfide. Now, these sulfate reducers also have a NIF gene. So while the plants are feeding these sulfate reducers, the sulfate reducers are producing the sulfide, which is reacting with iron phosphorus compounds to release the phosphorus, and they're also fixing nitrogen. And this all happens based on what these plants are doing. They release a lot of acetate when they're growing, and they make their own nutrients basically. And we have a little bit of acetate and DOC data. The DOC concentrations are seasonal, and based on what we have, they pretty much track the growth rates of these plants. And we've looked at the organic acids in here. Acetate is always really important, but also formate turned out to be kind of important. Not quite sure what this means. Now, these plants carry on glycolysis. They don't take it all the way to ethanol because ethanol is toxic. So they produce a lot of compounds that are great. And this is my last real slide. This is a teaser. These came out of these marsh organs this past summer. And if that's not elemental sulfur, I'll eat my hat. I haven't analyzed it, I don't know, but I would say that's elemental sulfur. The red stuff, what does that look like to you? And it's in the tissues of these rhizomes. Now, it could be anthocyanin, which would be boring, but I think it might be sulfate-reducing bacteria that are in these plants. Maybe. I don't know. I mean, we're trying to look at this from a molecular point of view. So I'm going to stop there, and I'm going to leave you with some conclusions. I've tried to convince you that the marsh possesses a number of stabilizing feedbacks, and this has implications for management. At low rates of sea level rise and or high relative elevations, the plants respond positively to an increase in sea level, increasing sediment trapping and soil biovolume, raising elevation. Organic production and mineral sedimentation are both important. Minerals more so at higher rates of relative sea level rise and low elevation. Organic production more so at low sea level rise and high elevation. Nutrient enrichment increases biovolume production, and Spartina appears to stimulate the production and availability of its essential macronutrients to meet its demands, but it is still nitrogen limited. And there may be important symbiotic relationships between the plants and the microbes. You know, there may be endosymbionts that are important, cable bacteria, or who knows what. I mean, there's something, I'm convinced that there's some really important relationship between the microbes and the plants that help to make this system work the way it does. And She Blinded Me with Science by Thomas Dolby, and Book It by the Tyson the music. And this would never have been possible without the help of these amazing technicians that I've had over time. Betsy Haskin was the first. She helped to design the whole synthesis routine that we use. And Karen is the latest and greatest. She's been working for about 15 years. She's amazing. She walks on water. Oh, and she won the first North American Marsh Organ race boat competition. And that's the end. So I'll stop right there. Thank you.